So just a quick introduction. Very nice to have Noah Mitchell from University of Chicago. He's actually physically in Cambridge, which is wonderful. So hopefully we'll get a chance to interact very soon once the blizzard settles down. And he's going to tell us something about the work that he and his group have been involved in the context of gut morphogenesis in the fly, if I recall correctly.
That's correct. Yeah, so thanks again for the invitation to be here to talk. And yeah, I'd love to meet up with anyone who also made it here on time if people are around. So yeah, what I would like to discuss is how biology uses physics to create complex forms. And I'm really interested in the mechanical programs that transform molecular and genetic cues into organ scale shape transformations. And you're seeing an example of that going on in this slide, where the midgut of the embryonic fly is breaking left-right symmetry and coiling into a chiral shape. And so that's what I'll want to focus on today. This is a question of morphogenesis, and while morphogenesis perhaps needs no further introduction to this audience, suffice it to say that in studying morphogenesis, there are these two broad themes which immediately emerge. First, that embryos become patterned with the expression of different genes and molecules in space and time. Here you see, for example, patterns of gene expression in the very early embryonic fruit fly. And the second is that there are these dynamic morphogenetic movements. And to study morphogenesis is to study the dynamics of this tissue geometry. And in order to bridge the gap from this sort of molecular and genetic lens to these organ scale shape transformations, we really have to understand the mechanical forces that drive the motion of these cells and these tissues. And so we're returning to this very old problem, as many are, to have a physical understanding that bridges these scales. And so in some settings, such as this early motion of the embryonic fruit fly, there's been quite an intense effort to formulate a physical understanding that bridges these scales, linking the tissue dynamics to patterns of cytoskeletal anisotropy and ultimately to genetic patterning. I'm really interested in translating that type of effort of taking 3D fluorescence data sets and converting them into physical fields and relating these observables to one another in order to build a physical understanding. I'm excited to carry that sort of lens forward into fully three-dimensional shape transformations like you have in your inner organs, such as in the cardiovascular system and the digestive system, which will be the focus of today. And so in that context, we have multi-layer tissues. Your gut and your heart are made of more than one tissue layer. And these are interacting, and so that adds to the complexity, and they're developing in fully three-dimensional space. So in this task, to have a mechanical understanding of this process, this is not trivial because, first of all, tissues can change shape via different processes. Cells can have pattern proliferation, pattern cell intercalations, pattern cell shape changes, area changes. But to solve that problem, there might be technical hurdles, as we'll discuss, but in principle, it's straightforward. You watch the material and find out what's happening. But as a deeper ambiguity is this many-to-one relationship between the patterns of mechanical stress and the constitutive relations of the material properties relative to mapping onto a particular choice that biology makes for the strain. And so in order to really address this, there are, of course, some tools that are emerging to try to measure stresses directly, but this is very, very hard right now. And so we rely on using perturbations and comparing our observations with models. So I'll propose that in order to sort of take this approach and study this 3D morphogenesis in inner organs, the Drosophila midgut is a really great model to make quantitative progress. The midgut, I think, really exemplifies some of the mechanical motifs of inner organ morphogenesis, starting from initially flat multi-layer sheets that wrap into a tube, fold into chambers, and coil into a chiral shape before ultimately generating physiological movements for digestion. And so here's what that looks like in the context of the embryo, of the fly embryo, with the midgut, the focus of our talk today, in blue, and the other digestive organs in darker blue. So my lab, about half of my lab, I guess, uses this model organ, and I think it's really great for studying each of these motifs in development. Now, this organ, just like your own small intestine, is composed of an inner layer, which is somewhat epithelial in character, certainly epithelial later on in development, and surrounded by circular muscle, here in yellow in this cartoon and false colored in this tubulin stain. And then overlying that circular muscle is a longitudinal muscle, which is a very thin meshwork, as you can see in these cartoons on the bottom. Can you see my my mouse, by the way? Yes. Okay, that's great. So this looser meshwork of red longitudinal muscle that's overlying the circular muscle. So I'll be using these terms throughout the talk of the endoderm, the circular muscle, and the longitudinal muscle. These are the three different tissue layers that form this organ. And so much of my group leverages Drosophila genetics and advances in microscopy to study each of these morphogenetic steps in turn. And I'd love to tell you about all of these projects from these different stages of development, but for time I'll focus on one which I think will do good service to the title of this conference. And that is on this breaking of left-right symmetry and coiling into a chiral shape. Now, this work actually began quite a while ago from discussions in Santa Barbara, particularly with Sebastian Streichen, as well as a bit with Boris Schraman. But there were other aspects of gut development that we needed to solve first. And actually, my initial measurements done there were honestly quite confusing. So since I moved to Chicago, we've been fortunate to make progress on this effort. And that's in large part by the people in the group. So we're quite a young lab, less than 2 years old, but I've had the great fortune to have some brave souls build up this story and other directions. And so in this story, particular, Nico, Chris, and Avi on the left have been sort of driving different aspects of the project from modeling, data analysis, and genetics. And I also want to flag that Nico Romeo will be on the job market this fall, and he's absolutely fantastic and has been collaborating with several different projects in my group as well as with other PIs at UChicago. Okay, and then I should just also mention that Emily Hendricks contributed some key experiments early during her PhD, actually during her rotation, and Fritjof Browns has been a collaborator on this project as well. So okay, so what's the question? The visceral organs in our bodies are left-right asymmetric, not just in their placement, like the heart is probably on your left, but also in their shape, their chiral shapes. And this is crucial for their function. And broadly speaking, this is for two reasons. The first is that we display left-right asymmetric gene expression early on in our embryonic stages, which then trickles down to the level of organ asymmetries. But on top of this, there's also a role of cell intrinsic chirality, where cytoskeletal structure of cells is intrinsically chiral, and the collective activity of these cells can generate organ scale asymmetry. And fly embryos, I think, are a marvelous playground for exploring this notion, since, as best we can tell so far, there doesn't seem to be any left-right asymmetric gene expression, at least not in these early stages. So we can isolate the roles of cell intrinsic chirality in organ coiling. And in particular, there are two unconventional myosin motors that are conserved determinants of laterality or chirality. And these actually break chiral symmetry at the molecular scale. So if you sort of sprinkle actin on a carpet of one of these, myosin 1D, it sends the actin in circles. And similarly, if you sprinkle actin on myosin 1C, it'll twirl the actin round and round. So I think this is really beautiful. It's also not terribly surprising that these molecular motors and can interact with actin, which is itself a chiral filament, in order to generate left-right asymmetry at the molecular scale. And what really I'm really interested in understanding is how this scales up from that level of molecules to how the cell becomes chiral, how tissues become chiral, and then how organ shapes become chiral. Indeed, these unconventional myosin motors are at play here, and that's been known actually for a long time, like 20 years. What you see, and just to demonstrate what I mean by that, if the wild type midgut coils as shown on the left, and it's been known for a long time that if you overexpress myosin 1C, so you just have extra myosin 1C motor in a certain with one particular driver, so in one particular tissue domain, then you can invert this morphology at the gross level. This is what has been known. And we can visualize that now here with new tools. I'll try to convince you that this organ's coiling proceeds via a sort of two-step modular structure. That there's a control at the level of a bias, molecular bias that will outline briefly. And then there's this sort of convergent extension-driven deformations of the tube that amplify this initial bias and canalize it into a stereotyped right or left-handed coil. So let's start at the molecular level. I'll just spend 4 slides telling you about a little bit of the, I guess you could say, the more biological component of this project, and then we'll move up towards morphometry and mathematics and addressing this issue. So the first thing I'll ask is, where is this myosin 1C acting to control chirality? Well, it had been known, again, for a long time that if you drove myosin 1C in this particular tissue domain called the brachyientron domain or bin domain, then this is sufficient to very often invert the midgut. So we thought, well, maybe the activity of myosin 1C in that domain biases the chirality right there. And so let's get rid of it. And so we looked at brachyientron mutants. This brachyientron is expressed in the hindgut, which is a different organ that's connected to the midgut. It's in blue in this cartoon. It's also expressed transiently in the outermost muscle layer of the midgut called the longitudinal muscle. And in these brachyientron mutants, these structures are all gone, or just about. There's small bits of them that remain. And yet the midgut can coil just fine without them. However, the chirality of that coiling is random. So the mechanical coiling program seems to be roughly intact, but the handedness is randomized. So we then ask, okay, well, which part of this domain is actually determining chirality? And the answer is just a touch subtle, so I'll say it in 2 parts. The first is that if we just think about, well, the only part of that brachyientron domain, which is actually part of the midgut, is the longitudinal muscle. So what if we try to kill off that longitudinal muscle? So we first targeted this tissue and just tried to make it very unhappy by expressing HID, which is an
No, I can ask a question.
Mm-hmm.
So how do I understand? So the circular muscles and the longitudinal muscles are they in the same tissue, or
they're different cell types? That the circular muscles are below the longitudinal muscles. So the longitudinal muscles are sort of on top. It's very much like your own anatomy with 2 different muscle types and then an inner layer. In your own small intestine, these are thicker. Each of these are thicker, but here they're just sort of monolayers of cells. Is that helpful?
Yeah, and they're intertwined with each other if they're on the same layer, or how do I, because the schematic you have here, it seems that they're really on the same layer. So
yeah, so they really are very, very close to being true monolayers. Certainly the longitudinal muscle is a true monolayer. They're not sort of entangled with each other or intertwined like this. It forms the way that you could think about the muscles is really forming a coordinate system. There's a circular coordinate system of sort of phalanx-oriented muscle cells that are all sort of side by side, and then on top of those is this very loose meshwork with lots of gaps of just strands of long longitudinal muscle. Does that work? Does that help? Okay. So if we make these cells pro-apoptotic, then we dramatically increase the number of embryos that have inversion of their midgut. And so it really seems like this longitudinal muscle is mediating the chiral symmetry breaking. I suspect that if we can really get rid of these cells more strongly, then this fraction would go up and up and up of inversion, perhaps to 50%. That's a bit speculative, but we're working on other ways of killing it off. So this seems pretty clear cut, that the longitudinal muscle is determining the chirality. We also wanted to check for sufficiency, whether myosin 1C activity in those cells it determines those chirality. And that was actually where the subtlety comes about. That normally these midguts are always the same chirality, and we'll discuss with some morphometry in just a moment. And with these myosin 1C overexpression, this is inverted. However, if you drive myosin 1C overexpression in midgut specific drivers, such as this one, or just in the endoderm, or just in the circular muscle, or in different parts of the circular muscle, or in all of the muscle, or in the yolk, or just in the longitudinal muscle, none of these can invert the midgut. However, if you drive myosin 1C activity in the hindgut epithelium, so if here are 4 different drivers which all express in the hindgut epithelium, then you get inversion. So what does this mean? It means that the hindgut epithelium, myosin 1C activity in the hindgut epithelium, is determining midgut chirality. So it's sort of a cross-organ communication, but that relay seems to be mediated by the longitudinal muscle. And so this actually, this seemed pretty strange to us at first, but in the end, I think it makes sense that it's because these two cell types were at a much earlier stage right next to each other. And so perhaps one signals to the other, then that longitudinal muscle goes and forms part of the midgut and determines its chirality. And so we're checking that hypothesis by now in a rather arduous experiment going to turn on extra myosin 1C at different stages, and we suspect that it will determine the chirality at this stage when they're still together back in the posterior. So this is about the extent of the biology that I wanted to, or the sort of mechanistic level that I wanted to put out there. Kankshi has a question.
Yeah, just a quick one. Are these muscles differentiated already?
Yes.
They're contracting.
Okay. Yes. So they are at this stage when the chirality is, when the gut is coiling, then yes, they are starting to now contract. So we see, and this, I'll show a couple of, I'll show just one video of this in the second part of the talk, but we actually have a whole project on the calcium regulation in these cells. So the circular muscle is contracting a lot, starting right just before coiling begins, and that I'll explain in depth. The longitudinal muscle also seems to have calcium pulses. This was not clear until we started developing an ex vivo culture platform and other things I don't have time to talk about. But they don't seem to be actuating any appreciable contraction.
Okay, yeah. The reason I was asking is it's more about the presence of the muscle than about those contractions, or not. Okay, it's the presence of the muscles.
Oh yeah. So in determining the chirality, you mean?
Yeah.
Yes. Yeah. My guess is, what I can say is that if the longitudinal muscle gets there, I think it's fine. If we for instance, we've done an optogenetic experiment trying to knock out its contractility. There's no phenotype, so it's not entirely clear whether it's working, but it would be, if that construct is working in those cells, then it's consistent with them not actually participating in the mechanics of the coiling. Does that make sense?
Yeah, it does.
And so, that's consistent with our brachioenteron mutants, which don't have hardly any longitudinal muscle cells. They still coil, right? So I don't think that the longitudinal muscle is really doing the mechanical active stress. we think that it's signaling to the circular muscle. And I'll show you a bit more data that supports that. And then the circular muscle's contractility is actuating the initial stages of left-right asymmetry.
Thank you.
Mm-hmm. So that's exactly what I want to talk about next. This is a great segue. This question where now we really have to invoke a lot of sort of mathematics tools to understand what is the physical process by which this organ is coiling. So now we know a little bit about the cellular determination. How is it doing this sculpting? And we can already imagine, I'll foreshadow that you could have left-right asymmetric contractions, or you could have a tube that sort of twists. I have my little prop here, that twists into the proper chirality through sort of active shear in the tissue surface. I'll tell you a bit more, but this list is are the sort of two possibilities because of some other kinematic constraints. And I want you to really just appreciate that these are biologically and physically very different. That in one case, left-right symmetry breaking is explicit, that the right cells are contracting more than the left cells, or vice versa. And in the other case, it's not. You still symmetrically pattern the activity, but what the activity is chiral. And so I want to just flag that these two possibilities coming up. And so which is it? All right. So to access the dynamics and the mechanics, we had to overcome some challenges. We have to image this organ deep inside the embryo. We have to create analysis tools for analyzing these complex geometries. And we have to relate their pattern surface deformations to the three-dimensional organ shape changes. So to overcome the imaging challenges, this is not particularly new technology. We use multi-view confocal light sheet microscopy with genetically cleared embryos. So what that means is that we sweep an illumination beam up and down and capture fluorescence in the transverse axis. And when we move the embryo through that, then we get roughly confocal resolution imaging of the midgut. But the sort of the far side of the embryo is still very fuzzy because we're looking deep in there and we're trying to image more than 100 and so microns. So what we do is we rotate and repeat at each time step multiple times. And we can take all these different views, fuse them together, and deconvolve the results into a consensus volume. So that gives us these panoramic data sets of the full multi-layer tissue, viewing the entire organ in 3D as it coils into these chambers. So then once we have these 3D over time fluorescence data, how do we quantify the dynamics of this coiling? Well, we want to understand how this relatively thin curved tissue is deforming in 3D. So here we use a level sets approach from computer vision to select a specific tissue layer and visualize how that tissue evolves in the surface over time. So this automatically captures the surface of the organ and tracks its evolution over time. And here I'm showing that visualized in the inner layer, but we can, of course, and will image other layers. And then to track the tissue deformations on the surface, we further invoke tools from differential geometry. Because if you have this sort of textured mesh in 3D over time, okay, how do you actually get a strain rate tensor out of that on the surface? So we're all familiar with mapping curved surfaces like the sphere to a plane to navigate around. We want to do something similar here, but it's more complicated because the surface is not so simple, and it's also continuously deforming. But once you have the right projection, you can start to think about how the tissue deforms in order to generate this shape change. And so what we do is we build projections of this tissue surface onto a Lagrangian frame. This has been published for a little while, so I won't go into the details unless it comes up in the questions. I'm happy to talk about it. And so here, even as the tissue changes shape in 3D, it's always mapped back to an image which is roughly the same. So, any given tissue patch doesn't really move around in the Lagrangian frame. And that allows you to track cells and extract kinematics about cell shape changes, cell area changes, cell rearrangements and tissue deformations. And this procedure also provides a sort of natural coordinate system defined across the entirety of the organ, which allows us to measure things like the center line of this curve and the surface strain rates that accompany its deformation. Okay, so now let's talk about those two things. From the center line, we can already quantify the chirality of the organ. The way that we do that is that we take little tangents on the center line, place them at the origin, and if they're chiral, they'll sweep out solid angles on the sphere. I'll say that one more time.
No, Noah, can I ask a question here?
Of course.
The center line is the geometric center. It's not the material center.
It is the material. I would call it the material center.
Because in order to get the center line, you have to draw a circle. And how do you choose or a shape? And how do you choose that unambiguously? It seems to me that you have to either label cells, and then you have to decide. Otherwise, you can't.
Great question. So what we do is at a particular time point, early on in morphogenesis, we conformally map our topological cylinder to the plane, and we have validated that at that early time, at least, that map preserves the direction of the circular muscle, so that
but conformal maps are too rigid. You cannot account for shear and you cannot account for eccentricity. So that already you've made a huge assumption there.
Right. So at I should just clarify. So at this early time point, this correspondence between the V direction in the UV plane and the circular muscle is quite accurate, I would say. At subsequent times, this fails to be accurate. There's lots of shear. And we follow those deformations. So we make a material coordinate system that follows those deformations over time. And we advect those initial hoops, if I go back to this previous image, with the motions of this tissue. Does this make sense? So then it's the barycenter of or just the mean 3D position of equidistantly sampled hoop, which forms our centerline point.
Yeah, but my question is, how do you choose the hoop?
Yeah,
maybe I miss.
Oh, that's okay. So I probably just didn't say it clearly enough. So if we look on the right, this cyan organ shape, at this time, if you look at its conformal map, then the vertical direction, like the V direction of the UV coordinate system of the pullback, is aligned with the circular muscle. So it is a material, it's faithful to the material's intrinsic coordinate system.
Okay.
And then if you advect that, then you stay in a material frame. I should be full disclosure, there's some error with that advection. You have to choose sort of smoothing parameters and things like this, but it's close. It's far closer than what you would get from a naive conformal map at each time point, for instance. Far, far closer to really tracing the material. And we've done some benchmarking of this, so I'd be happy to get into. Does this address your?
I think so. Yes. Thank you.
Great. So this is if we look at sort of small swept out areas on the sphere, whenever there's curvature of this tangent, then this is a measure of chirality. If you give a sign to whether this tangent is sweeping one way or the other way, then this gives you a direct quantitative measure of the sort of local, we call it local writhe. It's a kind of a measure of local writhe, quasi-local, maybe Nico would say. And asymptotically, Nico Romeo, this wonderful postdoc, showed that this corresponds to some other nice geometric features, which just for the cognoscenti in the room. And so if we use this, then what do we learn? If we look on the far right, we see that in our wild type case, or in the overexpression, which inverts the gut, that the chirality of the object is actually non-uniform along its length. So this thing is not just a helix, It's not just a kind of a funky helix, it's a perverted coil, meaning that it's right-handed and then left-handed, then right-handed, then left-handed. But on the whole, the wild type is mostly left-handed and is on net left-handed, and the inverted case is on net right-handed. So this gives us something to now explain, right? This is what I'm giving a sense of, right? That it's not simply that the gut is just twisting in one direction or bending in one direction relative to the other to give this coil, okay? And so what is it actually doing? We can go back and use these tools to not only look at the center line, but also the surface dynamics. So for instance, we can look at in-plane and out-in-plane velocities using this tool. And what after sort of thinking about it for longer than we'd like to admit, we realized that there are these two different ways of generating coiling via surface deformations. The first is that we have different amounts of shear on the two different sides of the tissue. And the second is that we take a tube that is bent. I should maybe make the movie of the first one play here. So the idea is that we found from these measurements before that this tissue is incompressible. And so I probably should have included a slide to that effect. But the punchline is that because we can't have differential growth, what we can have is differential convergent extension, that one side converges and extends more than the other side. And if the directionality of that symmetry breaking has an axis which coils around, then we essentially bend along a coiled axis. It could be that we bend this way and then this way and then this way, and that gives you a coil. And that's a very different thing, right? Because that's explicitly breaking left and right symmetry, where the right and the left are contracting and extending by different amounts or in different directions. That's a different thing than twisting a tube where the right and the left do the same thing, but you can sort of start with some other dorsal ventral symmetry breaking like this, and then you can take each little piece and cut and turn it so that it coils round and round. And that's the second simulation. Okay. So let's measure how much of these are at play. In order to do that—
Can I ask a question here?
Of course, yes.
If I integrate globally the shear induced by convergent extension, what do you get? I mean, in other words, what is happening at the ends? Because in the case when it's twisting, it's also shear. It's just a different kind of shear. In both cases, it's a shear mode. Yeah, but in one case, you are potentially rotating the end, and in the other case, you may not be rotating the end, the convergent extension. So can you elaborate? Maybe you're going to tell us about that.
I have a backup slide. There's some things on the backup slide which I haven't introduced yet, but maybe I'll just have you ignore them. So ignore the left and just look at the right. This is a movie of the posterior boundary condition. And you can see that this posterior boundary condition here, if you can see my mouse, is barely moving. In particular, it is not twisting. The same is true at the anterior. There's a bit of jogging on it, but there's no twisting. And so indeed, the boundaries provide topological constraints on this process. And the amount of twisting and writhing which sum to linking for those of you like Maha who know these things well, that is almost flat in this tissue. I think I even have Nico quantified this for us. And there's a lot on this slide, I know. But the point is that the twist plus the writhe is on the very right, with wild type in red, and this myosin 1C overexpression in blue. And yes, this is a very noisy thing to measure, but these things are not changing by very much. I mean, the scale here is very small compared to the amount that these are changing. They change by, pi over 2 roughly, and individually, but the twist plus the writhe is changing by 0.1, something like that. So this is very small. And so those geometric constraints are—
I think Vanessa has a question.
Very important. Yeah. Oh, Vanessa,
yeah. So it's just more clarification, really, because so the way I understand, just by looking at it, I'm not sure I understand if what you're saying is that there is a constraint to the convergent extension. So this tube does not change in volume in total, but it's becoming longer, right?
Yeah, it
And
doesn't change in
thinner.
its surface area much. The
Okay,
surface area, yeah.
so basically it's just becoming longer, doesn't have space to do anything, but and so it twists. Is that correct?
Okay. Yes. So you're jumping ahead, but that's where we're going.
Okay.
So I think with the caveat that the important step in the middle is that it actually bends. So this is the surprising finding that I'm getting to now, is that this initial left-right symmetry breaking is dominated by bending. And then
Right.
only at later times when we get to, I'll show this in just a slide, that
Okay.
the confinement and the geometric constraints that you guys are already talking about, you jump right to the punchline, amplify this initial asymmetry and actually, furthermore, we'll see, canalize it into discrete left and right-handed outcomes.
And so I was thinking when you were talking about the movements of the cells on the surface,
That's right.
you're thinking about those as causal or as an effect of
Great.
the fact
Great.
that this tube cannot do anything else and
Yeah. So I'm let me show you these slides to answer this question. If it's still
That's
unclear,
good.
we're right on it. We're right on those slides, right? So what we do is we track these tissue deformations on the surface, and it was helpful to sort of coarse grain this by thinking of there's sort of like a dorsal quadrant, a left quadrant, a ventral quadrant, a right quadrant. So this is what you're seeing in wild type and inverted. And then what we can do is construct a material frame from that measurement, which follows the tissue's twisting, which follows the tissue's bending in both dorsal ventral symmetry breaking and left-right symmetry breaking. And then we can measure exactly this question of whether we have bending dominated or twisting dominated dynamics directly. And so what Nico found is that the bending is turning on first and is quite pronounced. So what does that mean? What is driving? So that bending here is in black, and time equals 0 is roughly the onset of coiling. So the bending has already started to ramp up, and then the twisting turns on subsequently. So where is that bending coming from? A lot of it in this early stage, I mean, all of it essentially in these early stages, is coming from the asymmetry of these constrictions. So there's these 3 constrictions in the midgut, and I could give a whole talk just on the constriction formation, or more than one probably, because we've studied that a lot. But what I want to point out is just that if you look down the long axis of the gut, then even at a relatively early time, these constrictions are forming, and they form a little bit of a triangle when you look down there. So that's chiral. And then at the later stages, when there's both bending and twisting, that triangle gets bigger and bigger and bigger. Right? So that this chirality is amplified. So to address Vanessa's question, this is an active process, the formation of these constrictions that we've previously linked to the activity of calcium in the circular muscle cells. So there's these stochastic pulses of calcium activity that appear specifically at these constriction sites. And here's just a measurement of that and an example of zooming in with a traditional laser scanning confocal right at one of these constrictions. And so each of these constrictions have this localized calcium. And over time, this calcium gets bigger and bigger and bigger and less localized, as shown here in this space-time chymograph. So let's see. There we go. Yes. Okay. So that's just showing this again. So I'll return to this chymograph in a moment. I do want to say that if you turn off this calcium pulses here with a temperature-dependent, temperature-sensitive dominant negative allele of Cerca, then the coiling is halted. So there's no coiling here as long as the temperature is high. And then as soon as actually we let the temperature go back down, then coiling can resume. So clearly these calcium-mediated muscle contractions are part of this transmission from, so it's myosin 1C dependent activity in the posterior. It talks to the longitudinal muscle, and somehow the longitudinal muscle interacts with this calcium pulses to make these asymmetric contractions. So this is our state of understanding of this bending. It's driven by these initial asymmetries in the calcium-mediated muscle contractions. What about the twisting? Vanessa was just asking, is it active? Is it passive? Well, it's hard to test these things experimentally, so we turn to modeling. Oops, there's a small build problem here. In particular, the way that we're modeling this is at these late times, these calcium pulses start to broaden out, and by the later stages of coiling, they're actually spread out over the entire gut. And commensurate with that, we've been doing in a separate project, which I won't really get into, we've been doing some inference of what the stresses are in this organ. And in this calculation, you can find that the circumferential stress in this organ is also getting more and more uniform over time. What does this mean? It means that it's this convergent extension is not just driving these constrictions, but at late times is just elongating the tube, right? So if you have an elongating tube that's stuck inside this very confined body cavity, then maybe this could amplify these initial asymmetries, and the twisting could fall out as a passive consequence of that. This was the idea, and I think that I should just flag that this is the idea here is really that you have buckling instabilities, which are sort of amplifying this process. And the challenge for buckling is that buckling is instability. How is it controlled and stereotyped? Here we have something which is incredibly stereotyped, so much so that if you look at wild type and the inverted cases and you put them on top of each other with one of them mirror imaged, they lie on top of each other extremely well. So not just within each ensemble there is a strong amount of stereotypy, but even you have this sort of mirror symmetric developmental trajectories. And so could buckling really accomplish this? And I think our answer is that we're proposing yes, it can. That what we're suggesting is that there's early initial asymmetries from these constrictions, which bias you towards one developmental trajectory or another, and the bias is determined by this cell biology that we were discussing before, which we only partially understand. And that in looking at different mutants and different overexpression conditions, you will tune to bias one chirality versus another. So to really test this and hopefully show you that we think this is the answer, this mechanical canalization, Nico cooked up a home-built Kossera rod simulation engine. And if we start at a time when the gut is nearly, the center line is close to symmetric, and we put in the amount of convergent extension that this rod is feeling, then we get pretty good agreement with the actual shape dynamics. And this is ignoring any sort of anisotropic material constitutive behavior, no self-contact, reducing the effects of radius. So there's a lot of assumptions, but nonetheless, it works quite well.
Can I ask a question?
Yes.
So the Kossera rod,
yes,
has extension, shear, bend, and twist. So there are lots of parameters. And in addition, you have a natural curvature, natural twist, natural shear, natural extension.
Yes. So we're using viscoelastic behavior so that there are time scales effectively there are time scales associated with each of the plasticity, or I guess you could say the viscous behavior in which those reference bending and reference lengths are updated.
But how do you, there are so many parameters in the Coursera rod theory, especially when you have natural shapes which are not straight.
They're not straight.
It's not clear. Maybe you explained this. Sorry, I missed it. And how do you fit those parameters? There's so many.
I did not explain this. So what we, maybe, Nico could give you more of the dirty detail, but we did certainly sweep through each of these parameters until we found a regime where even in the absence of varying material heterogeneity, we have just visually decent agreement. And then around that, we really just did grids in these parameters. We didn't do any fancy ML or pin physics-informed neural networks or things like this, although I think there would be a value to that. And so this is a pretty insensitive regime. I mean, I could pull up sort of the tables of when we varied the different plastic timescales for the. I mean, it's really the relative timescales of there's sort of an elasticity versus viscosity component, right? And then there's the relative ratio of sort of twist elasticity and twist viscosity versus bending. And we treated the bending along the dorsal ventral and left right as being equal in all of our sweeps.
Okay, thank you.
That probably is just a partial answer to the question, given that there are many parameters. But we did our best to sort of, reduce down to a minimal set, and then just did grids.
Can I ask a question on? If I understood correctly, you said that the surface area is constant even locally.
It's roughly constant locally.
So the division is not doing anything. There is no extrusion here, and
there are no cell divisions. I could have said that early on.
Yeah. Okay.
So this is not a growth and form problem so much as a shape change.
It's very neat. I mean, is it because I think that if you're just incompressible, you're constraining a lot also the possibility of morphing, right?
That's right.
So is this something that the embryo is I don't know, is it just a biological constraint that helps constrain this possible looping and the other question is, in your model, how do you enforce this incompressibility? You just have a very high bulk modulus, or?
Oh, good. So in the rod model, I'll answer the second question first. In the rod model you're talking about, yes?
Yeah.
Yeah. So here we actually are not dealing with variable radii. This is because we weren't really sure how a variable radius would map on for a tissue, which is not a filled-in rod, would map on to the rod constitutive relation. I mean, I think it's not really clear. If you're wider but still thin, are you going to be easier? It can be easier or less. Is it like at first I thought, okay, well, if you have a constriction, then maybe it's like a hinge point, but it's not entirely obvious. There could be a lot of things changing biologically when you change radius. So we don't factor in the change in radius during these simulations. It's an oversimplification, and that's we think of as a blessing, right? So the incompressibility is really just a mechanism for elongation. That's what we're and the effect that change in radius has on the material properties we are choosing to ignore, just because, as Maha was saying, I mean, the search space becomes very high dimensional if you start having patterned.
Because I mean, I think intuitively it seems that if you're enforcing this incompressibility, it would make the space of deformations very frustrated.
That's right. Yeah, I think we have start of a little sort of just numerical project on this, thinking about in general, if we just have tubes that are incompressible, sort of what is the space of shape changes that you can get from that. I think this is a wonderful question, and it's sometimes quite. I mean, I could get into this, right? It's sometimes quite counterintuitive that You know, if you want to bend, well, then in certain limits, if the outer curvature you would normally think of as sort of growing relative to the inner curvature. Well, if it's elongating along the longitude, that actually means it's contracting more along the circumference, but it depends on the radius of curvature. So there's fun things like this where you get into corners where at least my own intuition is quite wrong. I'm sure some of your intuition would be intact. Okay, so I see I'm kind of going over, but maybe this is fine because it's all Q&A. Vanessa?
Yeah, I was thinking about this idea that it's incompressible. I feel like there must be luminal pressure in there, right? Because
Yes, that's right. That's actually how we're doing this this whole project. If I go back, oops, oh dear. So
But just to
sorry.
clarify, incompressibility is surface incompressibility. I think Vanessa is incompressible in the surface, not incompressible of the lumen is not incompressible. Lumen is compressible.
Right. And so how do you model a tube that has, because basically what you're saying is there is no change in the overall surface area, which is the endothelium, right?
There's very, I shouldn't, there's never no
sure.
surface area change, but there's very little surface area change relative to the shape changes that are happening in the composite tissue. That's right.
Right. But yet when you have this muscle that constrict, like these constrictions are kind of insane, right? So they're
huge.
They're very
enormous.
dramatic.
And you elongate extraordinarily much along the longitudinal axis as it bends. I can show you a picture of that if you want. So this incompressibility plus initial curvature, you just incredible strains on the circumferential direction.
So it really works like an inflated balloon type of thing.
And then, I think there, for small deformations, quasi-statically, it's a bit like an inflated balloon. That's right. I mean, that's a good analogy, particular for the way that we've been modeling the stresses.
And so what I was wondering is like, how do you think, because your experiment where you block the contraction of the muscles and then the whole process stops.
That's right.
Sort of suggests that also the elongation of the tube really needs this compression. And so intuitively I understand how you compress it in one point, then you make, a big balloon blob in the middle. I find it more difficult to think about how is it that compressing it this way, it's propelling the elongation of it.
Yeah, so these things happen in sequence. That first calcium pulses are very localized, and we see contractions, these convergent extension motifs localized to constrictions. And then subsequently, we see calcium everywhere, and concomitantly, we see reduction in radius everywhere. And so even in the chambers between the constrictions, the radius is going down, 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 and the longitudinal length is going up, up, up.
And so also the other muscles are starting to contract. So basically you have...
We don't know that. We have, without those muscles, this thing still coils. Without those muscles, this thing still elongates. It might not elongate by the same amount. We haven't measured that very carefully. But we just built a student, just a rotation student actually just made a heroic genetic cross to now have all 3 tissue layers imaged in this brachiontron mutant in light sheet. And so we're very much looking forward to imaging that, as soon as next week or the week after. Yeah. Akakshi also has a
no, I don't know if you had something to conclude, but there's also a couple of questions in the talk, in the chat box.
Well, I have just 2 slides. How could I
maybe conclude in 2 minutes?
Okay, so yes. So I'll just say that, if we just sort of blissfully ignore the obvious differences between, this very simple model and biological realities, what we learn from it is, I think, intact. That as you interpolate between a wild type or an inverted organ center line, which just initial left-right asymmetries, and elongate this thing in a box under confinement with these constraints at the end, then you're shape trajectories, your simulations dynamics in sort of a shape space here cluster into two stereotyped outcomes. And there is a splay in these developmental trajectories. And this is it's quite insensitive to the different ways that you sort of reduce the shape space. And so that's canalization. That means that the developmental trajectories are mechanically stereotyped and clustered into a right-handed perverted coil and a left-handed perverted coil, subject to these initial asymmetries that are amplified and canalized in this way. And this back to Vanessa's original question about whether the twisting is passive or active. What we can see from these very simple-minded simulations is that we get very similar twisting of this model rod without any active twisting, just bending and some initial bending and then amplification under confinement. And so this is the punchline of the talk, that we have myosin 1C dependent chirality determination in a different organ, in the posterior, in the hindgut, that through these longitudinal muscles determines the chirality of the midgut. We have left-right asymmetric contractions, which are calcium mediated, that calcium dependent, that bias the initial asymmetries, and then are amplified with growth or elongation of the tube, I should say, elongation of convergent extension, elongation inside the body cavity with geometric constraints. And that gives us mechanical route for amplifying these cell-determined chiralities and canalizing them into discrete outcomes. So with that, I have one teaser, which is that we also have this wonderful mutant that we're super excited about, where we can actually make two half guts, one out of the right tissue, one out of the left tissue. And when we watch this, then we see this explicit symmetry breaking happen right about now at the coiling stages. So this leads us to wonder, maybe the consensus that Drosophila doesn't have left-right gene expression. We would like to check this. So this is what we're up to now. And it could be post-translational, of course. So with that, I'll really give a big shout out, especially to Chris, Nico, and Avi, as well as Emily and Fridtjof. And I can take, I know I'm over, but I'd be happy to talk more. Okay, so there's some in the chat, you said. I'm not sure where the chat is. There we go. Oh, I don't see any.
I can read them out if it helps.
That would be great.
There's a question from Ashid about for these bending and twisting due to the buckling, the ends need to be anchored or fixed, right?
Yes. So, I think that there's a general question where you can have some freestanding tube and it can bend in 3D space and it can twist in 3D space without any anchoring. And there can still generate chirality. In our particular case, there are these additional constraints from seemingly fixed endpoints. We can just observe how much they move, and they don't twist. They move only, the posterior one's only something like 5 or 10 microns. The anterior one's a bit more, but it doesn't twist. And so that provides additional constraints that Maha pointed me to bring up earlier about the sort of link between twisting that happens, albeit passive twisting, we think, and the writhe of this organ tube, which is sort of like it's related to counting the number of crossings that you see of one part of the tube over the other.
Yeah, I guess related to that, I'll ask the second question from the same person, but just related to that, what's the sort of the main parameter here that's deciding twisting versus something else for buckling? Like, could it be like, Maha, I'm just thinking about like in the intestine, or there are other ways to have these instabilities that's not twisting, looping, or just those wave-like?
Yeah, I think you you can. So yeah, of course, maybe you're alluding to some of the really foundational work that Maha and company have done on sort of looping morphogenesis and in a different kind of midgut in the amniote. But this was not this was not chiral looping, and it was a set of perversions, right? So in some sense, I had chirality at the local scale. Sorry, I think I lost track of the questions. Yeah, go ahead, Maha.
No, I think the question was, are there other ways, if I understood Akanshi? Is that correct?
That's right. Like, does it have to be twisting in with these same constraints that you're mentioning?
Yeah, I see. So I think, the most important thing to say is that there could be differential growth in general. Right? The fact that we have this kinematic constraint that the tissue seems to the cells seem to conserve both their volume and their thickness, so therefore their area doesn't change much, right? That dramatically reduces, I think, the design space. But if you take, I've been using this tube as a bit of an intuition, but it really is a bit of a lie, because when you make it coil, then one side is compressed and relative to the other. This is differential growth, right? That one side can expand and the other side can shrink. And so if you can do that, then by all means you can shrink on the right side, and then the dorsal side, and then the left side, and then the ventral side, and you'll have a helix. So we have a more constrained sort of biological design space or shape deformation space to explore because of this the lack of that, or near lack of that contribution. So in that, in the absence of that, then we can have differential convergent extension, right, rather than differential growth, just different amounts of converging and extending in different axes across two sides. And we can have twisting, which is a different kind of shear, as Maha already alluded to.
So I know that we are out of time, so don't want to have people who would like to continue with other things to stay. But can I, with that caveat, can I ask a question, Noah? If I had a flat sheet and I had a metric which only had the non-symmetric parts, that means the single scalar, because if it's incompressible, then locally the trace must vanish. And then the only thing that's left over is the non-symmetric part. And if I remove identically the trace, I say not just the trace vanishes, but either I have only two components. So if I prescribe one component of the trace and one component of the off diagonal, I'm done. Have you or others thought about what? So this is kind of an attempt to address Akanshi's question. Have you tried to ask and answer the question of what class of potential shapes you will have if I had a flat free sheet, and then what class of shapes you would have if I had a tube which was essentially, for example, fixed at the ends corresponding to the gut and corresponding to the mouth and the anus, for example?
Yeah. So I haven't thought about this so much in the context of a flat sheet. But yeah, I have been asking this question of myself without really coming up with a good answer.
Because leaves can do this. You don't have to grow. You can just shear. You can just change the shear. Yeah. And leaf morphogenesis is, or not is, but it could be accommodated and perhaps some aspects explained using a similar idea.
Yeah. No, this is good. Yeah, I mean, like I mentioned, I had been prompting, someone in the group to sort of start thinking about this sort of shape exploration in this sort of the broadness of or the extent and the confines of this the kinds of shapes that you would get from this under this constraint in a more general setting. But we haven't had the time yet to really do justice to that question. yeah, been focused on trying to get us this particular case down to a T first. But we were very excited to explore more about that.
Thank you.